The overall goal of the following experiment is to characterize the physical, chemical, and biological properties of 6 biochars to be used as soil amendments for the remediation of contaminated sites. In basic biochar utility properties, percent moisture and organic matter, proximate and ultimate analysis, pH and particle size distribution are determined. In toxicant reporting, analysis includes seed germination and earthworm avoidance testing, as well as contaminant testing for polycyclic aromatic hydrocarbons, polychlorinated biphenyls, and metals including mercury. In biochar advanced analysis and soil enhancement properties, the biochars are tested for nutrients including ammonia, nitrate, nitrite, and phosphorus, as well as their ability to absorb or immobilize contaminants via evaluation of their specific surface area and cation exchange capacity. The results of these methods detail the physical, chemical, and biological characteristics of biochars and determine their eligibility to be used as soil amendments for the remediation of contaminated sites.
The main advantage of using these methods is that they are the same as those outlined by the International Biochar Initiative, or IBI, in their standardized protocol, and we have shown them to be accurate and effective at analyzing multiple biochars.
Many of these methods will be familiar to scientists specializing in soil research. Provided that the correct quality assurance and quality control measures are followed, these methods are reliable and easily applied to both biochar and their feedstocks.
In our previous work, we emphasized the importance of careful characterization of biochar prior to field-scale implementation. When we learned that the IBI was developing a standardized protocol, we had already begun this project, and so we were